Today we're going to be talking about ultra-conserved elements. And how many of you have heard of ultra-conserved elements? You've heard about it from me. You've heard about it from me. Anybody that's heard about it that didn't get it from me? Okay. Ask yourself why not, because they're fascinating. Ultra-conserved elements are elements that pieces of DNA that are identical in humans, rats, and mice. Why those three? Because those were the first three genomes that they had available. So they just ran them to see where they, matched. And there were over 400, about 481 different areas where the DNA matched between humans, rats, and mice. Now, you might, think about how you get there. And we're going to look at, first of all, there's an abstract from last year, perfect and imperfect views of ultra-conserved sequences. I'm not sure what perfect and imperfect views are exactly, but it's a reputable journal, Nature Review Genetics, and the abstract is available online. If you're part of the Loma Linda community or any other university community, you'll be able to get the entire article because, Nature Review Genetics, most libraries, university libraries, go ahead and get a subscription. And I'll just show you the abstract itself, and it'll give you some idea about what's going on. Wherever you see green, that's my comments. So I try to clearly separate what I have to say from what the article will have to say. Across the human genome, there are nearly 500, to be precise, 481, ultra-conserved elements, regions of at least 200 contiguous nucleotides that are perfectly conserved in both the mouse and the rat genome. No mistakes. No changes. Remarkably, the majority of these sequences are non-coding, and many can function as enhancers that activate tissue-specific gene expression during embryonic development. From their first description more than 15 years ago, their extreme conservation has both fascinated and perplexed. And we're going to go into why it would perplex researchers in genomics and evolutionary biology. The intrigue around ultra-conservative elements only grew with the observation that they are dispensable for viability. We'll go into that in a little detail. Here we review recent progress towards understanding the general importance and the specific functions of ultra-conserved sequences in mammalian development and human disease, and discuss possible explanations for their extreme conservation. Anybody need anything explained in that? Because it may be too technical. We're clear so far. If you have questions, feel free to ask. Now, this is the only, no, it isn't the only. There's another illustration that they have. But this is the most important illustration that they have, and you will notice that if you look over here, we have humans, we have some kind of monkey, probably rhesus, we have a rat, we have a mouse, we have a cat, elephant, chicken, and zebrafish. And you'll notice that not only are they perfectly preserved between the three species that makes the definition, for over 200 base pairs. But in fact, the rhesus monkey looks like it's identical also, or whatever monkey it is. And the cat looks like it's, except for here, pretty much identical. Not quite, but really close. The elephant has a few more changes. The chicken has a few more changes. The zebrafish is clearly mostly matching, but there are a number of changes in it. Yes?
Isn't it fascinating that cat and mouse seem to have so much in common?
Yes, that's an interesting point. Okay. Now, that's not the only definition you could have used. For example, they could have said 100 base pairs. And according to the review, for example, lowering the length threshold for human, mouse, rat comparisons to 100 base pairs increases the number of identified elements by an order of magnitude, that is, to more than 5,000. There are more than 5,000 areas where, for 100 bases, the human, the rat, and the mouse are identical. Okay. The problem has been known since 2003. That's when the first 3 genomes were available, and that's when they checked them out on each other, and somebody just, let's see what we can find. Now, there are possibilities for this phenomenon. And, okay, one is, could be just chance. Okay. What other possibilities? Anybody want to?
Some kind of foundational design principle.
Okay, there could be some design.
What about functionality? They're useful in nature, therefore they perpetuate over time.
Okay, what other reasons might there be for having identical base pairs?
Common origin.
Common origin.
Ancestry.
So we inherited them from a common ancestor, for example. That's one possibility. And common design. They're created by a common designer, or at least designer that knew how to copy somebody else's design. Okay. What other possibilities? There's one more at least that I can think of.
Adaptations that are necessary to survive on Earth?
Close. I think what people would say is convergent evolution. That is to say, the shrew-like animal that was the original ancestor didn't have it, but the rat evolved and the mouse evolved to have the same genome, and sure enough, it turned out that through lemurs and monkeys and apes, humans got it too. Not because the original shrew-like animal had it, but because whatever differences were there gradually decreased with time. They mutated in the same exact direction. And of course, you could have a combination of 2 and 4 to where it was almost identical, but there were a few changes, but they were the exact same changes. Can anybody think of any other possibilities? So I can't. I haven't read of anybody who can, or at least who said it. So I'm going to leave those with the 4 major possibilities. Well, what about chance? Well, the probability of getting the base pairs identical in the human and the rat, and we're just going to leave the mouse out of it for now, is 1/4 to the 200 power. Fairly straightforward. Which is 10 to the minus 120.4. Which I think would be highly significant as a p-value. It doesn't seem very likely to happen by chance. So leaving that out, we have common ancestor, we have common designer. We're going to try to avoid that if possible, because science, should be kind of methodologically natural, right? So, we just let's not go there unless we're pushed. Okay, and then finally, convergent evolution. Now, the interesting thing is, both the common ancestor and convergent evolution require something to select this and hold it there. Right?
Right.
I mean, because otherwise they would diverge, you would think. I mean, convergent evolution is just basically the target wasn't there at the beginning, but now is there. Whereas common ancestor would be the target was there all along, I think. Okay. Now, a few other facts to give you some idea of the difficulty of the problem, and then you will understand the amazement and the perplexity and all that are referenced in the article. The conventional divergence of mice from rats is 30 million years ago, and that of both from humans is 100 million years ago. Okay? Rats and mice have perhaps 5 generations per year. The shrew-like creature, if it's like modern shrews, it probably had 3 generations per year, just for what it's worth. So, maybe 3 generations isn't necessary. And besides that, rats and mice probably don't have 5 generations per year in the strict sense that every rat and every mouse has had 5 grandparents, great-great-great-grandparents within the last year. The reason being that when mom and daddy rats start getting babies, grandpa and grandma don't quit. So let's say 1 generation per year. We're going to be conservative on this. Okay? Now, there are perhaps 100 new mutations per generation. Most of them are neutral. If they were deleterious, they would be in trouble, and if they were good, you would start making new varieties of rats, which doesn't seem to be happening very fast. So they're at least near neutral. That means that they stay in the population most of the time. They don't die out. Okay, so let's say 100 mutations per year, 100 million years, that's 10 billion mutations. Simple math, right? Okay. Now, the problem is you only have a genome that's 2.75 billion bases long for the rat, which is the best case. The mouse is 2.5 billion for what it's worth. And that means that on the average, each base has mutated an average of 3.6 times. That means it's tried out practically everything, because there are only 4 bases you can mutate to. Now, of course, you have some back mutations, so that's probably a slight overestimate, but not that much. Basically, for practical purposes, every base should have been tried. Certainly as a mutation, and probably as getting, for a good share of them, getting all the possible variations. Now, think about how do you keep that thing pristine for 100 million years?
Maybe it's not 100 million years old.
Maybe it's not 100 million years old. Heretic. Okay. Maybe it is that they don't mutate. We're going to stick with standard science right now. So that's a possibility. And the review looks at that possibility. In addition to their strong conservation between species, ultra-conserved elements show reduced sequence variation within the human population. That is to say, you look at those 200 bases and they don't seem to be mutated as often inside of humans. In fact, that's one of the interesting things, is that if you were in manuscripts, you would call it the majority text. Okay. Upon their initial discovery, it was observed that human single nucleotide polymorphisms, SNPs, changes in one base, leaving the base in but changing what it was, known at the time, were heavily depleted by around 20-fold in these elements, further suggesting that mutations in these sites are selectively disadvantageous. So they thought, you mutate, you die. Or maybe you don't die, you don't have babies, something like that. However, some researchers speculated that highly conserved non-coding sequences could instead be mutation cold spots. There are hot spots for mutation, which mutate much more frequently than you might expect. We don't understand exactly why that is, but it does happen. And so that's a possibility. So maybe if there are hot spots, there are cold spots, and so these ones are protected from mutation somehow, presumably resulting from a theoretical molecular mechanism that repressed mutagenesis. Subsequent larger scale human population sequencing confirmed an overall depletion of variants in human ultra-conserved sites. That is to say, yep, there are not as many mutations, but also showed that variants are found are skewed towards those with rare derived allele frequencies. That is, when you do find it, there's one over here and nobody else has it, and there's another mutation over here and nobody else has it. Or maybe there's a small population that has it. That's why the majority text is, in fact, the majority text. And so it showed the variants are skewed towards those with rare derived allele frequencies, and there's a figure that shows that. In other words, common variants are particularly depleted in ultra-conserved loci. It isn't that there's a mutation where 60% of the population has it and 40% doesn't. Or maybe even 10% has it and 90% don't. It's like 1/10 or 100th or 1,000th of a percent that have that particular mutation. Whereas extremely rare variants occur at levels close to those observed elsewhere in the genome, excluding the possibility that ultra-conserved elements are generally protected from mutation. So that means, yes, it can mutate. And no, it's not, it's not just that it's being protected from mutation. So, well, let's see. The other possibility is maybe if they mutate, you die. Or at least you don't reproduce, or at least you reproduce at a slower rate or something. It needs to be a lot slower, because if it's like 1/10 of 1% slower, Selective pressure just isn't good enough to find that. So, functions of ultra-conserved sequences, that's our actual title of this part. Human rodent ultra-conserved sequences are reported to play a role in various aspects of gene function and expression regulation, from activation of transcription to exon splicing. So they do have functions, and maybe if you change it, the function doesn't work. Consistent with the original study, 23% of ultra-conserved elements overlap exons of protein coding genes, whereas the vast majority, 77%, do not. So you can kind of understand if you're coding for a protein and you change it, the protein won't fold right, or it won't do the enzyme function it's supposed to, and that'll be a problem. But 75% don't even do that, which means that it's a little difficult to understand why their exact sequence should be that critical. Some ultra-conserved exons are alternatively spliced as poison exons, but those are the protein coding exons, which interrupt the reading frames of genes, for example, by introducing an in-frame early stop codon, and target the resulting messenger RNA for degradation. In other words, you make the messenger RNA, and the body goes, wait a minute, that's a stop codon, that's not going to produce anything, let's just get rid of it. Now, of course, it doesn't actually do that. There's no brain thinking about that. But there are enzymes that do the job anyway, without thinking about it. And so, yeah, there's maybe a function there. But the function seems to be to destroy the RNA in that case. Functional dissection of ultra-conserved poison exons revealed them to be crucial for cultured cell growth. So this is the ones that code for proteins, or actually don't code for proteins, but should, so to speak, with some exhibiting tumor suppressor activity. So the coding genes were going, well, maybe we understand why some of them are being preserved. Although preserved that well? We highlight their importance in establishing normal development while having no obvious impact on viability, and we investigate possible explanations for their perfect sequence conservation. So they're scratching their heads, how do you keep this thing perfect without killing everything off? If they don't influence viability and they don't influence reproductive prowess, then it seems a little difficult to figure out why they should be preserved. Okay? Surprisingly, even a substantial mutation rate of 5%, nearly half of the enhancers remained active. In one extreme case, an ultra-conserved enhancer showed residual tissue-specific activity upon mutation of 20% of ultra-conserved base pairs. That seems to say you can mutate this thing a lot without affecting viability. These results indicated that ultra-conserved enhancers do not commonly lose their enhancer function upon even significant levels of mutation. It's worse than that. Somebody did some experiments where they snipped out the entire 200 base pairs. Just joined the rest of it together, and mouse doesn't have any of that at all. Remarkably, mice that were homozygous null, that means they had both chromosomes with missing, I don't want to call it gene because most people think of genes as coding for protein, but base pair segments, whatever you want to call them. For individual ultra-conserved enhancers, or if they're males, it's an X-linked factor, and they only have one X chromosome, so they're missing that entire testing for the only chromosome they have that could give it to them, in either case, show no indication of increased prenatal lethality. They don't get reabsorbed as embryos. Were fully viable at birth. They were born live and lived well into adulthood. Why do they say we lived well into adulthood? Because at a certain point they killed them and looked at the insides and couldn't find anything wrong. Assessments of pathology, growth, and gene expression did not reveal obvious detrimental phenotypes. That is, they looked at, they couldn't find tumors that they were growing on them. They couldn't find that they were growing too small or too large, although a few of them, they were a little bit small. But, small rats don't necessarily all die off either. Resulting from the loss of these enhancers. So it didn't seem to matter. Given the extreme sequence conservation of these loci, the lack of apparent phenotype was quite surprising to many in the field. What is going on here? And led to various hypotheses as to the reasons for the observation. And believe me, if the article could have found what the hypothesis was, they would have told you. They summarize currently unanswered questions remain about why ultra-conserved sequences are so highly conserved and exactly what function evolutionary selection is acting on to maintain this conservation. To date, half of all ultra-conserved enhancers deleted in mice have not been shown to result in a potentially detrimental phenotype. The fancy way of saying, that's a fancy way of saying half of them, you can't tell anything is wrong. Okay? And there has been no direct demonstration that loss of any ultra-conserved enhancer results in reduced viability, fertility, or fecundity. They not only live just fine, they have babies just fine, and the babies seem to be just fine as well. There is nothing wrong with these mice or rats. Now, the question comes back. How does one perfectly preserve 200 plus base pair segments for 100 million years without making sure that the errors are not reproduced? Well, maybe they're not errors, but that's the problem. If they're not errors, then why shouldn't they be reproduced? Now, let's go back to our explanations. You may remember we looked at those questions before. We did a little math. Now let's do a little more math. Okay, well, let's say that there was... it's hard to envision evolution happening within the last 6,000 years. But it is not that hard to envision a creator acting within the last 6,000 years. 7.5 thousand, 10,000, you can do the math for any of this. If you have 6,000 years, you have 600,000 mutations. That's about 1 every 4,600 bases. You divide the genome by that and you get, and let's say we're going to be even more conservative. We're going to say 5 generations per year. So we're going to multiply that whole thing by 5. Again, that's probably not completely fair, but that makes a mutation every 920 bases. That means that you have a 200 base pair segment, and you have a mutation on this side, a mutation on that side, but you just missed that one. Which of those two scenarios looks like it is more likely?
Second.
Anyway, comments and questions. Some of them appear to be in brain development. Others appear to be in kidney development of the ones they've studied. Of course, they haven't managed to study every single one of them exhaustively. But the ones that they have, that appears to be the most likely reason for them. But again, apparently there's a large variation in how many mutations you can have in any one of them. Okay, the answer is the only thing you get is these expressions of surprise. Now, one thing to keep in mind, this is a review article, which means more than likely it was invited, and more than likely there were reviewers of the review article as well saying, oh, you missed this, and you need to add that, and you didn't get this one quite right, and they go over that before they publish it, which is a strength of peer review. But they don't actually mention the difficulty with the mathematics. I'm mentioning it because I know enough about human reproduction, and I looked up what rat and mouse reproduction are, and they're in the same ballpark as humans. Well, actually, there are a lot of mechanisms that defend the genome from mutation. It's just that they're not 100% reliable. There are mutations happening in your genome every day. Most common one probably being cytosine deaminating and turning into uracil, which behaves like thymine in this setting. And so all of a sudden it matches with adenine instead. And so when you unzip it and reproduce it, then all of a sudden you've got an adenine instead of a guanine on the other side. And that happens all the time. And there are enzymes that go down. Jim, you can correct me if I'm wrong on this, but there are enzymes that go down and detect deaminated cytosine and replace it or put it back on or something.
Yeah, DNA repair is so important because you have to have a fidelity rate. In other words, you have to make faithful copies of your genome with such extreme accuracy that after going through quadrillions of mutations in your body every day, maybe even quintillions, depending on if you live next to Chernobyl, the cells are so good at fixing those mistakes that you might get one mistake out of every 1 billion or 10 billion base pairs. A recent paper came out to look at how many enzymes are involved in just finding and fixing mistakes in your DNA, and they use artificial intelligence to help find a couple hundred that they didn't know about. There's at least 605 proteins involved in just keeping your DNA in pristine condition, error-free, without mistakes. But the one mutation he's talking about can't be detected, because this is like, say you're typing a letter and you meant to put cat, but you typed in bat instead, and the spell check doesn't pick that up because bat's a real word. And this is the same problem with these enzymes, is that, hey, this looks like a normal base, there's nothing wrong here. We didn't know it got switched from a cytosine to adenine. So over time, you have less and less G's and C's and more A's and T's. And if there was really that much time, you would expect there would be this continuous decline in the number of G's and C's in our genomes because they can't get fixed. They're not recognized as mistakes.
That's a great point.
So if you were going to share this with other people and you wanted to come up with an analogy, This is, imagine like a shooting range, and a whole bunch of people for years and years and years have been shooting at these targets, and for some reason there's huge patches of the target that don't have a single bullet in them. And it's like how there is no explanation for how you could have these cold spots in the genome that aren't crucial, yet somehow have dodged 100 million years of mutations that didn't get fixed.
Well, there would be a possibility if there was some kind of pillar in front of it. So that as you're aiming at the target, you hit the pillar instead. But what would you do in this particular case? You see, there is a particular mismatch repair. Once the cytosine is deaminated, it no longer matches guanine very well. It's a mismatch repair machine that just looks for mismatches.
As long as one of the strands is intact, because they can look at the opposite
That's
side.
correct. Well, the odds are you're not going to mutate both bases simultaneous.
It's much less rare.
You know, then the question is, if there is a mismatch, how would the repair machine know which of the two bases needs to be corrected? And the repair machinery actually looks at, well, which was the original, the vetted copy, and which is still questionable that we're checking on. And this kind of thing is going on all the time. Problem is, if both sides have been vetted, you can't tell which one is which.
Yeah, well, the other thing is that if the repair mechanism doesn't figure it out before it's time to split,
Right.
then you just miss it.
But there is another repair mechanism that compares the sequence to the other chromosome. It's called recombinant repair system, and figures out which of the bases is wrong that way.
Compare mom's.
In other words, it will compare, because we have most chromosomes in duplicate, the odds that the same base would go bad on both of them simultaneously is not so strong. So by comparing, you identify which base is the misfit, and that machine can repair such problems.
Even with all that repair, studies have shown when you compare the genes of parents and grandparents to yourself, or do this in animals or cell culture, that typically in a generation there's about 100 mutations. That's where
I think 120 is what it is for humans.
range is. Like if you have kids really young and eat a plant-based whole food diet and stay out of the sun, and you're not living next to a uranium mine, you might get by with passing only 20 mutations, 25. It could be upwards of 300 if you're a smoker who has kids later in life.
Yeah, measured rates that I have seen have varied from 60 to 180. But you're probably right. If you pick the right people, you might get it down lower than that. You might pick the wrong people. You know, if you live around Chernobyl, it's probably more like 300.
Yeah, John Sammer told me he's seen like 300. The guy who wrote Genetic Entropy and the Mystery of the Human Genome. And this is true in multiple species too, not just humans. They found this in mice.
Now, to be fair, rats and mice are a little bit less than humans, and that's why I didn't go with 120, which is kind of the consensus figure. But
They also make their own vitamin C, so you got
but it's not that much less per generation. Well, again, I'm a little bit cautious. Certainly from this data, I can't tell you it's 6,000 versus 7,000 versus, but certainly, in the under 10,000 range is probably reasonable. And it suggests that you don't go millions, let alone hundreds of millions of years, or you couldn't do this. Yeah, it's still chance, but just fewer chances.
Yeah, okay.
It's as if the range has only been shot at, 20 times versus 200,000 times.
But even if chance plays an important role, then still it's difficult for the evolutionary theory to explain the origin of such a complex system. Those are molecular machines acting with some sort of their own mind or something like that. So it's so complex. So that is one of the things that is so difficult to explain from the evolutionary perspective, right? Like how the origin of information in the first place is there.
Well, that's a whole different subject. For example, there are 600 known protein coding regions in humans that don't exist in chimpanzees or gorillas or anything else that we know of. How do you get 600 brand new genes? This isn't modification of chimpanzee genes so that, this is like the whole thing.
Right. But then if we analyze all this information as from the designer point of view, so chance is not a player here, but a designer, then it's probably easier to explain that maybe there's a purpose for this highly conserved sequence that the designer may have lived there for us to discover, as many other discoveries are taking place, like on Earth.
Well, I don't know that that purpose is enough of an explanation. I think you have to put recent purpose as well. I mean, I guess you could say a designer sat over the rat mouse creature and over the creature evolving into a human and made sure that no changes were done here, no changes were done here, although it's not clear as to why that designer would do that, because you're protecting it for millions of years because it's critical, but it's not critical.
But the question that I have there is, has this information been measured in all cell types? Because, from organ to organ, you have different, even though these conserved sequences are kept in different tissues, they may have played different roles because the expression of genome is different for every tissue.
Well, here's the thing about that. In fact, other than in the immune system where it's routinely changed deliberately, most of our DNA is in fact very, very close to identical. You might have 120 changes per. You might have probably 1,000 changes in skin cells, for example. But that's 1,000 spread over 3 billion. So that's like 1 in a million changes. That's not very much change in terms of percentage. It's literally 1/10,000th of a percent. So, the cells that eventually produce ova or sperm are relatively protected, particularly ova. They grow to a certain amount and then they stop. And they stay stable for the lifetime of, in this particular case, the woman. For guys, they do have to continue producing sperm, and so one of the things that happens is the older you are as a guy, the more likely you are to have mutations in the sperm, and you just hope they're not too bad. So get married to a young guy. No. Of course, then again, his, you're getting his mutations plus his daddy's, so I'm not sure it makes that much difference. But the important thing is, I mean, this is obviously a small part of a very large topic. And we can go on, discussing the related topics for a long time. But it's a small part that I took a poll nobody had heard of. And I hadn't heard about it until I read it. What?
It's fascinating. I love when we can tie math to these things because statistically speaking, it's so improbable that it's just ridiculous.
See, this is an important part of the creation evolution debate. And yet you don't hear about it. And you don't hear about it because most creationists haven't actually run into it. And people who believe in long ages aren't helping them.
Right.
Because, you don't air your dirty laundry in public. But the fact that there is dirty laundry bothers me a little bit if I'm going to be somebody believing in long age. The website is named after the book that I wrote. It's called Scientific Theology, so the website is scientifictheology.us.